Sì, sì, okay, perfect. Okay, good morning, everyone, and welcome to this seminar. We are today in company of a dear friend, Dr. Jairo Mendoza Roldan, which is working now with a parasitology group of the University of Bari in Italy. And so they are very good friends. And today we will speak of a very interesting topic, because many of that, like me, doesn't have so much or at all experience in reptiles, and sometimes with parasite of reptiles. But as we will see with Dr. Mendoza, they are involved also in many zoonoses, and we would like to, with him, to deepen into this field. So please welcome Jairo, and good lesson to everyone.
Well, thank you so much. Hello, everyone. I will start the presentation. Just 1 second, please. So hello, everyone. First of all, happy April Fool. I would like to thank Professor Antonio Barcasia and the University of Sassari for the invitation to talk about, well, my favorite subject, which are reptiles, and of course, their parasites. So as reptiles, it's a very diverse group of animals, given their species, morphologies, and physiologies. Likewise, their parasites are also their parasites and their parasite diversity is very, with a lack of a better term, gargantuan. So if I were to come here to do a presentation regarding systematics and taxonomy of the parasites of reptiles, I think it will get very boring very fast. So what I will try to do today, I will focus on key point topics of which I've been working the last 8 years within the herpetological parasitology, which are ectoparasites of reptiles, vector-borne diseases of reptiles, and parasites of reptiles of zoonotic concern. So just to get started, let's talk about the class Reptilia. Reptiles, actually, the word reptiles comes from the Latin repere, which means to crawl. So it seems that the former taxonomists, what they tried to do was to group a very diverse group of animals within the same class. And in fact, when we see the different morphologies and the molecular evolutionary traits, as you can see in this phylogenetic tree, we can see that actually they are very, very diverse and very different one from another. In this case, for example, you can see that the group of the lizards and snakes, which is called Squamata, is not so related to crocodilians and dinosaurs to birds. So as we are having more information regarding the molecular evolution and reconstruction of the phylogenetic relationships, we can see that in fact we have different groups of reptiles that are not related or have very different morphologies and physiologies, which we can have to take into account to be able to understand the parasites they can harbor. So the main groups we can find are turtles, which are the most diverged at first, and actually we still don't know what is the actual origin or the ancestral type of turtle. Then we have a big group called Lepidosauria, which are all of the real scaled reptiles, and we have snakes, which is Serpentes, suborder Serpentes. We have Sauria, which are the lizards, and then we have the living fossils Tuataras that are from New Zealand. But we also have the Amphisbena group, which is like a combination between snakes and lizards. It's called blind snakes or two-headed snakes, but it's actually a group apart. And finally, we have the crocodiles, which are the ones related more to dinosaurs and more related to birds. So what in all this sense, we can see that reptiles are a very successful group of animals, and they have changed very few in the billions of years that they are here in Earth. And each day and each year, new species are being described as we are being able to go to different geographical areas And so we have more than 1,000 species of reptiles, which are within 102,000 genera. So they are divided in three main groups, as I mentioned before. We have the Lepidosauria group, which is the widest and which the most number of representatives. And you can see here we have the living fossil, which is the Tuatara, which represents an order called Rhynchocephalia, which only has one species in New Zealand. And then we see all of the other species that belong to the order Squamata. So we have all of the type of lizards. This is the Amphisbaena I was mentioning before. And then we have all the different kinds of snakes. On the other hand, we have the group Testudines, which represents the animals that are marine turtles, freshwater turtles, and tortoise, land tortoises. And this is a very small group, which still the origins is not well known, but we know that there are two orders, Pleurodira and Cryptodira. This depends on which the way of the head is inserted inside the carapace. Finally, we have Crocodilia, which is a small group, that has maintained practically the same since its origin, which represents all of the type of alligators, crocodiles, and gaviales. And it's a small group of 26 species. But what can we say is that, as I mentioned, this diversity, it's very difficult sometimes to actually say what is a reptile. How can we define this so diverse group of animals? So this is a very simplified slide, which I show you some trait or characteristics of which a reptile could have or may have that they share with the other representative of reptiles. So let's go one point to the other to see actually that there's always going to be an exception to the rule, and that there's not actually one certain characteristic that we can say, okay, this is a reptile. For example, we have that they are cold-blooded or ectotherms. We have that they have all of the teeth of the same shape and size, that they lay eggs, or they are oviparous. And when they are born, they can take care of themselves after they hatch or after they are born. And finally, that they have a tough and dry skin, which can have scales or plates. So let's go one through the other to actually see the situation we have. Is that, for example, they are reptiles are cold-blooded. In fact, they are, and they are ectotherms, which means that their body temperature depends from the environment, and they have different ways of gaining or losing heat. For example, radiation, convection, evaporation, and conduction, which is very important because the animal needs to move between a term that is called POTS, preferred optimal temperature zone, in which the animal goes and gains or loses weight to be able to do its metabolic process. It's important to remember that reptiles cannot digest beneath 7 degrees Celsius. But if in fact, so they cannot generate heat. So if we do, or if we have an infrared image of a reptile, what we would see is that they will have a coloring which is in the colder spectrum, as you can see in the snake, which compared to the hand of the human and of small lizards. But in fact, recent studies have shown that, for example, this is a species of tegu lizard in the southernmost part of South America, meaning Argentina, Uruguay, Brazil, in which this animal in the colder periods or in the winter can in fact generate heat. So even with more studies, we see that animals have different traits that we cannot put them all together. Afterwards, they have, we can say that all reptiles have the same teeth shape and size. This is in fact almost true for all of the animals that have teeth. Remember that turtles do not have teeth, they have some type of beak. But for those that have teeth, in fact, most of them have the same type of dentition. For the exemption of snakes. And snakes, we can use the way that they have the dentition to be able to classify them in 4 groups. So we have 4 groups of different fangs, which are the Aglyphas, that are snakes that belong to Boidae or Colubridae families, in which they, as you can see, they have all of their teeth as hooks, which enables them to catch the prey and be able to grasp it before killing it. Then we have Opistoglypha, which means that they have one little fang in the posterior part of the maxilla. And this is for some colubrids. As you can see, colubrids can be Aglypha or can be Opistoglypha. This means if they have this little fang in the posterior part of the maxilla, they will need to grasp and bite the prey twice. This teeth is connected to a special type of gland that is called the Duvernoy gland, which is a modified salivary gland belonging only to the colubrids and produces a less potent venom than the other venomous snakes. Then we have the real venomous snakes, such as Proteroglypha, which are all of the elapid snakes, meaning real cobras, coral snakes, and mambas. And they have a fang, which is not more, it doesn't move, and it works like an hypodermic needle, which it's connected to the salivary gland, which is modified to be a venom gland, and they produce mainly neurotoxins. And finally, we have Solenoglypha, which are the real vipers, and these are the ones that have a large fang in the anterior portion of the maxilla, which is recovered by a connective tissue and works as a hypodermic needle that they can move, and also connected to the salivary venomous glands. So as you can see, snakes do not have this characteristic of being all of the teeth at the same shape and size. Remember also that most of the reptiles, they can lose their teeth continuously as sharks, and they can recover them. So it's pretty normal to find, for example, when you do a corpological exam, you can find some fangs of the reptile that you're examining. On the other hand, we have different reproductive strategies. So in fact, not all reptiles lay eggs, most of them do. But for example, if we see the reproductive strategies of snakes, we have that some of them are viviparous, which depends on the level or grade of mammotrophy or placentation. And then we have those that are real oviparous. And the snakes that are real oviparous, they belong generally to the pythons or colubrids. And instead, those that are viviparous are mainly those of the vipers or boid snakes. And there's one example of one trait that most of the reptiles, or if not all, have, that is that they can take care of themselves after hatching. Why? Because reptiles are precocious animals, which means that they are born already with the morphology and the form of an adult, just smaller in size, and they grow through the years. So this is something that is true, and as you can see, these vipers that are being born right now, they already are animals that can defend themselves and can find prey and are already having venomous functional glands. So now I'm going to show you a video that maybe you have seen already. It's from the documentary Earth from the BBC, narrated by David Attenborough. And with this video, I want to show you two things: the predation between reptiles, which is important because they can, as they eat one to another, they can transmit different parasites from one to another, and also the possibility of animals that are born, they already have to do everything alone. So this is an example in the Galapagos Islands where newborn of marine iguanas are being predated or are being hunted by colubrid snakes of that region. So just to show you also that reptiles are stoic animals, so they don't show their signs of illness or stress. It's very difficult to read sometimes reptiles. But okay, continuing with the different traits, the final one is that they have tough, dry skin with protective scales or plates. Generally, those that have scales belong to the squamata or to the Lepidosauria order, which are the real scaled ones. And those are mainly lizards and snakes. And they have different strategies in the sense of being able to shed the skin. So they are the ones that truly shed the skin, in which the lizards, most of them do incomplete ecdysis of the skin, in which they can sometimes eat also the skin. And snakes have to do a complete ecdysis of the body. This is important because if a snake doesn't do the complete ecdysis, it enters in a type of pathology called dysecdysis, and this can be produced by parasites, mainly ectoparasites. On the other hand, we have the other two groups which actually do not do a proper molting or shedding of the skin, which are turtles and crocodiles. And in this sense, they have a type of osteoderm, which is a combination of the scale or of the skin with the bone. And for example, as you can see in the crocodiles, we have different scales that actually have bone, and a part of it, it's skin. So reptiles, this richness of species of reptiles and diversity of reptiles is distributed mainly in tropical and subtropical areas. This is because, especially because of the temperature conditions. And if we go further on into the Mediterranean basin, we can see that the northern part of Europe, of Africa, has a wider or a larger richness of species, but Italy is between one of the groups considered with a high biodiversity of reptiles still. In sense of richness of species, this is a graph from June 2000, and this shows you that the different number of species are mainly from the neotropical area, the Ethiopic area, and Asia. But as I told you, among the years, new species are being discovered. So from last December, you can see that the number increased. It almost sometimes doubled in some areas, and in Europe we have more than 60 more species described. So every moment species are being discovered, and this is important because some parasites are very specific of their host, or reptilian host, and so when we discover a new species, for example, the tuatara in New Zealand has a type of hepatozoan which is transmitted from a specific type of tick. So talking about the diversity of parasites, this is just to mention the groups that can infect or infest reptiles. I will focus on that are in bold, because as I told you, it's a wide diversity with different morphologies, systematics, and taxonomy. But they are divided into big groups: the single-cell parasites and then the Metazoan parasites. So we have the phylum Apicomplexa that is divided mainly in two groups, which are the order Amasporida, which is represented by the Plasmodium parasites, which are also the malarias, but these are not zoonotic or pathogenic malarias. And it is believed that the origin of the mammal malarias is from the malarias of reptiles millions of years ago. Then we have the group Coccidia, that's a subclass Coccidia, which is divided into groups, the Adelorina, which one of the representatives mostly found and very easily found in reptiles is Hemogregorines and Hepatozoon. As you can see here, this is Gamont. But it also contains different type of Hemogregorines as Hemolibia and Cariolysis. Then we have Emeriorina, which is divided in three groups: Cryptosporidae, that we find some species that are specific for reptiles and pathogenic for reptiles. And then there is a possible, as I will show you later on, there are possible species that are from mammals that could be transmitted by reptiles. Then we have Sarcocystidae, which were represented by Sarcocystus, which most of the species are from mammals, but there are some species that are being described from reptiles, which could have a zoonotic concern. And finally, we have Emeridae with some species such as the genus Isospora. Then there's another clade, which is called excavata, divided in three superclasses, which are Fornicata, that has Giardia. Giardia, generally, it's not pathogenic for reptiles, and most of the species are not zoonotic, but there are some assemblages of Giardia detected on reptiles, which I will mention further on. Then we have Parabaseala, which has the Monocercomonas. And one superclass that's very important that I will discuss at the end is Eoglenozoa, which has two important of zoonotic concern parasites or protozoarians, which are Trypanosoma and Leishmania genus. And finally, we have Amebozoa, which the species that is, it contains the Entamoeba from mammals, Entamoeba stolytica, but the Entamoeba from reptiles is called Entamoeba vivax and can produce very severe clinical signs, but it's not zoonotic. Regarding the Metazoan parasites, most of them are not of zoonotic concern. So those that you can find in both, some species have some implications, but most of them are very specific to their host, or they can be transmitted by predation of small reptiles from mammals, carnivores, especially. So we have in the Metazoan parasites the platyhelminths, which are divided into Nematoda or flukes, and cestodes or tapeworms, which I will emphasize or focus on one genus, which is Spirometra, that is the causative agent of Sparganosis, which is an emerging disease of not only reptiles but other type of wild animals. Then we have Lophotrochozoa, that it has a very important group, which is Acanthocephala, that are called also thorny-headed worms, and these are easily found in the necropsy of small snakes and lizards with no zoonotic concern or damage to the host. So most of these species that I've mentioned before and in this slide, actually they have some type of, not saying symbiosis, but some type of homeostasis with their hosts. So they don't produce in normal numbers or a normal parasitic load. They will not produce any type of signs or deleterious effect on the host. Then we have the also in Ecdysozoa, which are the ones that the parasites that mold their skin. We have a very big group called Nematoda, of the Phirum Nematoda or roundworms. And here we can find different families or orders that are very specific to reptiles. So the important representatives of reptiles that are very commonly found are, for example, from the family Rhabdicidae, the genus Rhabdias, which are lungworms, found most likely or most commonly in snakes. And then we have Strongylidae and Strongyloides, which are also in the pet market of reptiles or wild animals. They are very common findings in the crocodilogical exams, but they seem to not produce, and maybe in herbivorous animals, they can be of benefit to be able to digest cellulose, for example, in iguanas. Then we have the group Spirurida, and in this group, the ones of importance, not zoonotic, but importance given their transmission strategies, is the group Philaroidea, which has different genus, for example, Foleyella and Osvaldofilaria. And this group is transmitted most likely by diphtherians, mainly mosquitoes, and the microfilarias are sometimes found in the blood smears, and the adults live in the subcutaneous, and that thus can produce very strong deleterious effects or even death of animals. Foleyella is very common from chameleons, especially those that are, for example, captured in the wild and then sold in the reptile trade. So the animal comes looking normal or looking healthy, and then with the stress caused by all of the movimentation and the transportation and the new environment, diminishes the immunology, and then this causes the animal to be able to start to manifest clinical signs. Also in Nematoda, sorry, just a second. In Nematoda or roundworms, another important group is Ascaridida, which the species, the genus most important of Opistoglypha found in important group is Ascaridida, which the species, the genus most important of Opistoglypha found in important group is Ascaridida. Which the species, the genus most. I'm having a return of the audio. Okay, sorry for the disturbance. Okay, so we have the group Ascarida, which, for example, some important genus are Ophidascaris that are found mainly in neotropical snakes. But from some studies we perform in Brazil, they seem to do not have a deleterious effect with a high parasitic load of the adults in the gastrointestinal tract. And then we have Oxurida or pinworms, which the eggs are commonly found also in wild traded animals or synanthropic animals in the cropological exam. Finally, there are two groups in the Ecdysozoa which are very important, and I will discuss in depth with which are the class Arachnida, subclass Acarina, which are mites and ticks. And this is the group of which I have most experience working on the ectoparasites, taxonomy, and also in the vector-borne diseases. And then we have the Pentastomida, which are similar to or are more related, are arthropods related to crustaceans, and they are also classified as an emerging disease produced by foodborne or water contamination, specific from snakes, and can infect other animals such as mammals. So with all of this context or introduction, I've been working the last 8 years, 10 years with a special area of medicine, veterinary medicine, which is herpetological medicine, which is the area dedicated to ectothermic tetrapods or reptiles and amphibians. And in this area, as you saw, we have a very diverse load of different type of patients and also of different parasites. So for me to come here and try to explain or talk about every type of parasites will be a very difficult endeavor. So my idea for today is in the concept of herpetological medicine, I will discuss of three main topics, of which are linked in my experience to which are the parasitology, given by the different zoonotic parasites that are related to reptiles. All of this under a One Health approach, in the sense to better understand how the interactions between human and reptiles can lead to emergence of different pathogens or to the distribution of parasites. So the reptile-human interaction in this paradigm of One Health is given by different types of interactions, such as, for example, pets, which actually is one of the industries of trade with more success, given that the increase on the reptile trade and reptiles as kept as pets. And then we have the food source or trophic relationships between humans and reptiles. And finally, one of the areas I've been working more, which are the synanthropic animals, which means that animals that generally we can find near urban or peri-urban areas, such as geckos or lizards or snakes mainly. And this interaction, especially synanthropic interaction, is being given by the anthropic pressures on the environment, producing environmental modifications. So in this case, there are three specific or important environmental modifications given by anthropic pressure, which are deforestation, urbanization, and fragmentation. So these figures, what they are showing is Brazil. There is a type of biome very important that is called Mata Atlântica, which is in the southeast and south part of Brazil. And we can see the degradation and fragmentation of this biome, mainly because the major concentration of urban areas in Brazil are especially from the states of Rio de Janeiro, which you can see here, and Sao Paulo. So the urbanization and population of these states is one of the biggest, which has produced that animals that were considered forested before, that used to live in forested areas, have take advantage or can take advantage of the urban conditions, which is called synanthropy. The sense that animals come to the urban settings and can profit from the heat, from different animals considered as pests, for example, rattlesnakes to rodents, and have all of the conditions to be able to thrive. So this synanthropy or the interaction between wildlife and humans, given that they are very near fragmented areas, and also this is not only the case from Brazil, we can also see this in anthropic areas such as Italy, but increases the encounters and accidents with domestic animals and humans. This will then increase or will then finish or conclude in the increase of diseases transmission and especially parasites. So in last year, we studied the transmission, or we did a review article called Zoonotic Parasites of Reptiles, a Crawling Thread, in which we discussed the different transmission pathways parasites can have from reptiles to humans, and pointing out or highlighting the different diseases that can be produced by specific parasites. So we single out or specified in this figure 3 types of transmission, which I will discuss some of these diseases that they can produce from each of the pathways. And each one of them is associated to one type of interaction. So we have the first is by foodborne parasites given by the in different regions. The herpetophagy is something that it's very normal or it's part of the culture, which means eating reptiles and most importantly raw or uncooked meat from reptiles. And in this case, as I was saying, sorry, herpetophagy is something very common, and it's mainly considered something from Africa or African or Asian countries. But in fact, what I'm showing you here is from northern Colombia. Some regions, they eat iguanas or freshwater turtles, and it's something considered in specific settings near the forested areas, something common. Such is the fact that, for example, there are some regions that iguanas are called tree chickens. So as just to demystify that only in some countries people eat reptiles, and as for example with amphibians, it is also very common to eat the toads. So herpetophagy is something very intrinsic of different cultures around the world. Those foodborne parasites, or those are parasites that can be transmitted by eating, are specifically or transmitted by eating uncooked or raw meat. Generally, if the animal is properly cooked, there will be any issues. And they are divided, or they are represented by 5 groups, which are the flukes, the nematodes, the protozoans, cestodes, and pentastomids, which are also called tongue worms because of the shape. And in most of the cases, or almost all of the cases, humans are not intermediate hosts or not final hosts. They are just aberrant or dead end host for which an accidental transmission has occurred by the ingestion of the parasite. And then this is the reason it will show different clinical signs depending on the parasite migration or localization. So this is a table from that revision in which we distinguish the type of parasite, and then we also mentioned the geographical area and the implication or the importance of these parasites. In this sense, the importance can be firstly cestodes, represented mainly by Spirometra, as I mentioned before, then pentastomids in the African and Asian continents, nematodes, specifically, for example, Angiostongius cantonensis and Trichinella, and then with a low degree of importance, or in fact, because of scarce studies in this matter, we have protozoans and flukes. So I will mention, or I will talk to you about two important parasitic diseases that are Sparganosis and Pentastomiasis. So the first one is Sparganosis, which is produced by Spirometra, a genus of cestodes represented by different species such as Spirometra mansoni, Ranarum, Mansonoides, and importantly in Europe, Erinaceropei. And these species are distributed worldwide, and the cases are more focused, or are more, the outbreaks are more centered in the southeast of Asia, given the cultural behavior of eating uncooked meat of snakes and amphibians. But what happens is that generally the carnivores are the final hosts, and then they excrete the egg, which then turns into a coracidium, which goes into the water, and is ingested by copepods and turns into a procercoid. Then in the intermediate hosts, which are fish, snakes, amphibians, and others, they turn into plerocercoids, and then normally they will go to a carnivore, ingests the intermediate host, and then the cycle goes. So humans can be infected by contaminated water or by ingesting the different intermediate hosts. This is just the other example by the CDC, but it shows this exactly the same process. And this cycle is focused or was from a recent study this year of a revision of Spirometra cases in Europe, in which we can see that we have different wild animals that have been reported as infected as intermediate hosts. And most importantly, as you can see here, cats may be an important, have an important role to bring or distribute and spread the Spirometra in to the urban settings or near humans. This is because cats, as you know, feral cats and feral dogs, and also cats that are kept housed cats, mainly can have a part of their life in outdoors, and this has an important effect under the fauna of local fauna, because cats can eat almost all of the animals. So this is an example of a cat eating a snake, which is one of the most important intermediate hosts in Europe. And in fact, some years ago, the first report of Spirometra confirmed molecularly from Spirometra erinaceropei was from a snake, a water snake, very common, called Natrix natrix. And then the study that I was mentioning, which is the Sparganosis in Europe with molecular data, you can see here that in Italy, most of the cases are northern to central Italy, and some have also been reported in Sardinia. In this case, snakes seem to have a very important role as cats. And Sparganosis is an emergent disease that is very important because it causes various symptoms depending on where the Spargana or the larvae go throughout the hosts, in this case humans. So we have, for example, if the larva migrates to the eye, you can find ocular Sparganosis. If it goes to the neurological system, it can cause neurosparganosis with very non-specific signs or symptoms. For example, headaches, weakness, seizures, abnormal skin sensation, which is also as a creeping disease, and numbness and tingling depending on the nerve affected. And there's one very important manifestation or form of Sparganosis, which is when it's produced by a big intake or a large intake of larvae, which is the prophetic Sparganosis, or called also Sparganum poriferum. And this type of Sparganosis produces various lesions under the whole body as the larvae migrate throughout the host. On the other hand, we have a very important group of parasites, which are the Pentastomida. As I mentioned before, Pentastomida are actual arthropods that the systematics is not well known, but it seems that they are related to crustaceans. In this sense, we have Linguatula serrata, which is hosted mainly by dogs. But from the reptiles, we have three important genus, which are Armilifer, Porocephalus, and Rayetela. So Armilifer is the one that is most reported as cases from human cases. And the first time I saw actually the Pentastomids was in one field trip in Brazil, in southeastern Brazil, near Sao Paulo, in which we found a snake very weakened, dehydrated, almost agonizing, with open mouth respiration, which is a clinical sign of the respiratory disease. And after a couple of hours, the animal died. And when we did the necropsy, we found in the lung this type of vermiform parasites attached to the lung wall. And this is after seeing this for the first time, I saw, or it was evident that this is not something uncommon. This is another Botrops jararucusu, or lancehead viper, in which the animal started manifesting clinical signs, and then the adults, which are these, started coming out from the glottis connected to the trachea here. And recently, from this video from, this is a bushmaster snake, Lachesis muta, the biggest viper of South America. And also it had this type of parasites coming out of the mouth. So in fact, the Pentastomids produce a very large or very big deleterious effect on their host, on their main host, but also on their intermediate hosts. And this is because the definitive host, mainly those from reptiles, the invasion or the localization or tropism for the lungs makes the snakes a very sensitive target, because most of the snakes have just one lung, a functional lung. So if the lung starts to, and then the functional part, which does the gas exchange, is very small in comparison to the whole organ, which has the lung, functional lung, and then the air sac. So that portion, which is very irrigated and very small, is where the parasites prefer to localize and fixate, and they occupy the complete part of the lung, and the snake then dies of asphyxiation. So the definitive hosts of Pentastomids are reptiles in their most of them, and Linguatula serrata mammals, mainly dogs. Intermediate hosts can be other mammals, such as mainly rodents for snakes. So that's why vipers are most of them, vipers and boas and pythons are the ones that are more affected, because they have a diet based mostly on mammals and rodents. And then we have also, in this case, intermediate as aberrant intermediate host humans. So the morphology, I will talk afterwards of the life cycle. For the morphology of Pentastomids, they have most of the species have a sexual dimorphism. You can find here the larger one is the female and the smaller one is the male. And then they mainly attach, as I told you, to this part of the lung, which is the functional part. And we have different life stages, such as the larva. As you can see here, the larva is the representative that looks more like a crustacean or an alien. And then we have different stages of nymphs, 9 to 10 stages of nymphs. So these are the two infective stages which will produce clinical signs on humans. And this, you can see also the larva then transforms or changes its conformation after the migration. And the eggs, this is very important because generally one of the ways to do the early diagnostics of the infection in reptiles is through coprological examination, and then we can find two types of egg, which are the mature or immature. Sometimes we can see the larva inside the egg, so this is very important to know. Also, we can see the parasitic load. So how does the life cycle of these animals or parasites work? Well, the adults, as I told you, they are on the lung. So this is a clear image of the lung, and you can see is that it's in the conformation of the reptiles, mainly snakes, lung, and lizards, is like a beehive, which you find the different portions, and the functional unit is called faveoli. And this is the area which they prefer because, as you see, it's very highly irrigated. The adults remain in that area, and then they produce the eggs, which then go to the digestive system and are excreted. So the intermediate hosts, generally they have or eat the eggs or eat the larvae, depending on the where they eat or where they obtain the infection. And then the larvae or the eggs eclode inside of the intermediate host, and they go to different parenchymatous organs such as liver, spleen, and the mesenterium. There they will await until they can reinfect another snake or another reptile. So what happens with the humans? There are very different ways that humans can be infected. Or the main one is by eating uncooked or raw meat of snake. Then the other one is by handling animals infected and not having proper biosecurity measures, or also with contaminated water where the reptile was before. And the human acts as an aberrant intermediate host, in which, depending on the number of larvae or eggs that will migrate, for example, if he ingested one larvae, or one egg, the larvae will migrate, and the person will have just one larvae in liver, spleen, or mesenterium. But if he ingested a very large load, which is normal, the normal thing to happen, they will distribute around the different organs, even in the lungs. So Pentastomiasis, as I told you before, is a neglected and emergent disease, and it can be produced in reptiles by mainly two genus, which are Armilifer, different mainly from Africa and Asia, and Porocephalus, which is distributed mainly in the neotropical region till the southern part of United States. So this is a table which the main species, which are Limotula serrata, which I will not discuss because it's mainly from dogs. And then we have the different Armilifer species and Porocephalus. The main ones are Armilifer, Armilatus, and then the Moniliformis, because they are distributed especially in the areas where human beings or people have, by culture, they eat raw or uncooked meat, or even, for example, do some type of healing process with the raw skin of the snakes. So it's very difficult to diagnose this disease in humans because generally it depends on the parasitic load, and most of the cases the person will not present any clinical signs, and most of the diagnostics are made or post-mortem, or when the person is doing another type of exam and they perform an X-ray and they see in the X-ray the larvae. So the regions where this mainly occurs is, as I mentioned before, is Africa and Asia, and it's because the popular behavior or custom of eating raw or cooked mainly snake meat, and most of these cases are asymptomatic. But then there are other cases in other geographical regions that show us that this is a disease that can be detected by the same reason that we have a lot of most of the cases, they do not present clinical signs. So this is a paper from Brazil, from Mato Grosso state here, which a patient having abdominal pain was found with a type of lesion of granuloma, which you can see that had still viable pentastomid inside. How he got infected, it's not very clear yet. Probably not by eating a snake, but by having a contact with a contaminated water or environment. And then, as I told you before, the only proper way, because there are no molecular or direct diagnostics, mainly all of the cases are diagnosed post-mortem. This is a dog, and this is a person, which you can see along the different larvae. So in these cases, most likely the animal or human will have presented some clinical signs, but in most of the cases, the person goes unaware of the infection. So the pentastomids, as I told you, they migrate to specific organs, but this doesn't mean that they cannot migrate to other locations inside the host. This is, for example, Linguatula serrata in the eye of a patient, and the transmission of these parasites, as I mentioned before, is by contaminated water where the animal was exposed, or by improper handling. So we as veterinarians, we need to be aware of the risk of handling animals without proper measures, because we don't know, for example, if an animal was brought from an endemic area, if in fact it still has or has had the infection. So the clinical signs, as I mentioned before, are generally asymptomatic, and it will depend most likely on the organ which was most affected. And also the clinical signs can be produced when the larvae, at given a certain time, they can endeavor or thrive for some couple of months or even years, but there will be a moment that they will die. When the larvae and nymphs die, they can also produce some type of deleterious effect or clinical signs, depending on the affected organ. Finally, from pentastomids, we can also consider other species that have been scarcely studied. For example, those species that are from lizards or geckos. And there was a report long time ago in the early 30s, of a suspected case of creeping disease. Creeping disease is produced by Rayetella midactyli, which is a type of pentastomid that is associated to geckos, and the intermediate host are cockroaches and other insects. And it was believed that the man was affected by Rayetella midactyli because he had ingested raw geckos sometime before. Is there a real risk with Rayetella? In my opinion, yes, because we still don't know the whole distribution. For example, this is a case of Rayetella in Tarantula mauritanica, or Moorish gecko, which is a gecko that is from Mauritania, but it's now distributed in whole Europe, and some cases in some places of North America. And we found with a high parasitic load of eggs, mature eggs in the ecopharmacological exam. And this is the risk given by, can be given by animals, domestic animals predating this specific species of synanthropic lizards, which are very common, synanthropic geckos, or the human contact without a proper handling. Finally, from the foodborne pathogens that I will discuss, we have Sarcocystidae or Sarcocystosis, Sarcocystis, Actually, most of the 200 species are mammalian associated. They are the two most important are Sarcocystis hominis and sui hominis, which the humans are actually the definitive hosts. So the infection for humans, it's through the ingestion on uncooked or raw meat, and then the whole cycle of the Sarcocystis is happens inside the host, the human host, which excretes in the feces the sporocysts. So generally, this doesn't involve reptiles at all, and most of the cases are in Europe, and it can produce diverse clinical signs depending very linked to the gastrointestinal system, depending on the level of infection. Nonetheless, I bring this parasite today to discuss. It's because some years ago, 2017, some outbreaks of a mysterious illness started to happen in Malaysia, in which there was, for example, a case of a Marine soldier that went to have his vacation in Malaysia and then returned, and he started having some clinical signs with different type of non-specific symptoms such as fatigue, fever, muscle weakness, and nothing else that could say that he had contracted some type of specific disease. So, of course, the differential diagnoses were a myriad of diseases that can produce these non-specific signs, such as viruses, bacteria, and also trichinella was also taken into consideration, but it was negative. So also toxoplasma was considered in a moment because of the different unspecific clinical signs. So after different diagnostic techniques, it was detected or identified that it was actually the causative agent of this type of these specific outbreaks was Sarcocystis nesbiti. Sorry, this type of Sarcocystis or type of species, it's believed to be associated or have as definite host snakes. So what happens actually, not the ingestion of snakes or raw meat of snakes, but the contamination of water by feces of snakes, which could lead to the infection on humans that will act as aberrant or a dead-ending host. And then, as in other mammals occurs, the sarcocystis will develop inside the skeletal or cardiac muscle. This is very important because the clinical signs, obviously, as they affect another type of systems, will be very different from those that are specific for other species of Sarcocystis. Something very important is that the how they focused or how they believe or enclose that the species associated to Sarcocystis nesbiti are snake was after they found molecularly species that eat the intermediate hosts such as monkeys and rodents, which are mainly big snakes in Malaysia as pythons and cobras. So this was, in this sense, the foodborne pathogens. There are others, but I just focus on those that have major importance, but are also some way or somewhat neglected. On the second hand, we have, and also sharing with Pentastomiasis, the transmission pathway given by contamination or the environment, by feces or the contact with the animal. And this is mainly through animals that are kept as pets, as human, with the human interaction, or synanthropic animals. I will mention two parasitic diseases, which is cryptosporidiosis and giardiasis. But before, just to let you understand that the pet trade or reptile kept as pet is becoming increasingly more popular, and it's very common in areas as countries as the United States or continents as Europe to find in all of different pet stores. You can find different type of reptiles, from a very small leopard gecko to a large reticulated python. So the number of animals, of reptiles kept as pets has increased in the last decade. Just for instance, in Canada, there are more than 164,000 snakes. And in the United Kingdom, you can find that we have almost the same number of 400,000 animals of lizards and snakes kept as pets. And this is tending to increase. This is also another type of recent graph showing the different districts or regions of the United Kingdom, in which the number of pets, of reptile kept as pets, is quite high in this specific context. So the zoonotic parasites associated to pet reptiles are mainly those that can be transmitted by the direct contact with humans and reptiles, and they are in the protozoarians and ectoparasites. Ectoparasites, they are also in between, as well as protozoarians, they are also in between the synanthropic animals, which, as I will show you some moments after, the parasitic load in synanthropic animals can be quite high with no deleterious effects. So for Cryptosporidium, which is protozoarian, very common for snakes, there is an important species which produces grave clinical signs, or very important clinical signs in snakes, is Cryptosporidium serpentis. And this is important because generally when a group of snakes is managed, or a collection of snakes is managed together, detecting in the quarantine moment or beforehand that the snake is infected is pivotal to be able to save or to prevent more infections. This type of Cryptosporidium survives long periods of time outside the host, and by the management of one snake to the other, they can be transmitted. And the most pathognomonic or important clinical sign they produce is the gastric hyperplasia. So the snake, which will be with certain diameter, you can see where the stomach is, as apparently it ate something or has a large ball. And the problem is that animal cannot properly digest and will produce the death. There is no, generally, when animals are infected, it's advised to separate it completely from the others. There are some treatments which are not very effective. So most of the cases, or the animal dies, or euthanasia is advised depending on the level of infection. But most importantly, some species of Cryptosporidium have been detected that are from mammals, zoonotic, have been detected in snakes and other reptiles. For example, Cryptosporidium muris, Tizeri, and Parvum. But in snakes, the importance of detecting other Cryptosporidiums needs to be very seen with caution, because snakes feed most likely or mostly on rodents, which then you can find if the rodent was infected with a type of Cryptosporidium, you can find the Cryptosporidium in the snake feces. Differently, for example, recent studies in Italy have shown or showed that lizards such as geckos and turtles were infected with oocysts of zoonotic or potentially pathogenic Cryptosporidium species to humans. So it's important because they do not eat rodents. So this needs to be further addressed to see, in fact, if they can be have a role or be important reservoirs of other Cryptosporidium species. On the other hand, Giardia Protozoas have been found just in one occasion in reptiles, specifically those of zoonotic concern, and they were found in Spain in this short communication paper in two types of species of lizards, the Iberian rock lizard and the Bocage wall lizard, which are lacertid lizards, very common and synanthropic. And they were found with assemblages associated to livestock and those of zoonotic concern. This is very important because these animals live in sympatry near human settings or where livestock is kept. So there can be some type of transmission. And likewise, further studies should be are of importance to see if it's a wide distribution or if it's a local situation, depending on the degree of synanthropy of these lizards. Wait a second. And finally, and when the transmission pathway that I will focus more is the transmission by vectors, because of two reasons. First, the vectors are important ectoparasites, arthropods, that they are divided into important groups, which are mites and ticks, and then we have Diptera represented by mosquitoes and sandflies. So this is associated mainly to synanthropic animals, which will have the importance with the human interaction, but are also possible to find in pet animals kept as pets that have been brought from a facility that harbor different animals, so the transmission of ectoparasites it's common, or that they have been brought from the wild or from a wildlife setting without a proper quarantine. So first of all, regarding the acari or acarina of the herpetofauna or ectothermic tetrapods, this is a wide diverse group also, as all of the parasites are reptiles. So we have more than 500 species in 61 genera and 13 families, but they are classified mainly into groups, which are Acariformes, with importantly the order Thrombidiformis, and then Parasitiformes, which is divided into main orders Ixolida, which are the ticks, and Mesostigmata. So Thrombidiformis, it has different families that can affect specific reptiles, as for example, Pterygosomatidae and Harpyhincidae, which are very species-specific ectoparasites, are mainly from the squamata or lizards and snakes. Cloacaridae, which are mites that live in the cloaca of turtles, marine and land alike, and are transmitted venereally. They are mainly from only from turtles and specific from turtles. And then we have Thrombiculidae, also called chigger mites, which the larva is the only parasitic stage, which can infestate not only reptiles, but also some species, humans. Within the Mesostigmata, we have most of the families affect solely snakes. For example, Omentorella is a species specific with one species from Africa. Ixodorhynchidae and Heterocerconidae are common South American acari from snakes. And Entonicidae is a very important and curious group because they are lung mites. So they are harbored inside the lung, in which the information we have of this, if this mites is very scarce. Finally, and importantly, the group of mites that has the most veterinary importance is Macronicidae, represented by the genus Ophionisus, which is from a mite that can feed on reptiles, snakes, lizards, turtles also, and also from humans. From ticks, we have two families, mainly the Ixolidae, which are the hard ticks, and the Argacidae or soft ticks. And there are mostly in Ixolidae, we found very different genera that can infest reptiles, but they are also most of them can infest also humans and other mammals. So you have Amblyoma, where most of the species that infest reptiles are distributed in South America and Asia. Botrocotrum, which is mainly from Australia. Dermacentor, that is distributed in different ecoregions. Amaphysalis hyaloma, with one very important species from turtles. And then we have Ixodes, which there are different species, but those that affect reptiles are distributed mainly in the Palearctic and Nearctic regions. From the Argacidae, we have less information, but thus far, we know that two genera can affect or infest reptiles, which are Argas, and most importantly, Ornithodorus, which has had different new species described in the last 2 or 3 years, mainly from northeastern Brazil. So what are the morphological differences to be able to distinguish this myriad number of species of mites and ticks? Well, one of the most important things that we can see is the respiratory stigma, or the distribution of the respiratory system of mites. That is one of the easiest way to distinguish or to identify properly this type of mites. So we have Thrombidiformis. The main group which affects and has a parasitic role is within Prostigmata. So prostigmata actually means that the stigma and the tracheas are located within the natosoma, which is the mouth part, and then the first cocci, as you can see here. And mesostigmata differently means that it has the stigma located within the third and the fourth cocci. And Ixodida, or the ticks, generally the stigma is located in the lower part of the idiosoma, which is why they are called, formerly they were called metastigmata. Also mesostigmata, differently from Ixodida, which are in the same group, Parasitiformis, they have a specific type of setae, which are called Tritosternum, which is located ventrally of the natosoma, and it works as a sensory organ, which allows the mite to be able to localize and to move and to properly move throughout the space. And then, as I mentioned before, the stigma is located just in the third and fourth pair of cocci. Other than that, something very important is the reproductive strategies, which Mesostigmata has many, and Ixodida mainly they are oviparous, but Mesostigmata, they can lay eggs one at a time, or they can be viviparous, and then as you can see here in this type of Exodorhynchidae mite, you can see the larvae formed inside the female. Other very important aspect that we can separate easily mites and ticks is that ticks that are larger than mites. You can see here, this is Amblyomma humerali, a very common reptiles tick, and this is our Opistoglypha natris mite. So this is an adult, and obviously ticks are bigger. And also ticks have an organ, a sensory organ in the first tarsus, which is called the Haller organ. That as a stigma allows them to be able to move and to sense the different atmospheric pressures or different stimuli from the environment. So mites and ticks normally in most of the occasions or infestations, they will not produce deleterious effects on the host. But as you can see in this picture of Morelias pilota or carpet python from Africa infested by botryocoton ticks or guana ticks, this is a very high parasitic load. So in this case, this animal will indeed suffer from direct deleterious effects. And these deleterious effects from a high parasitic load can be given, for example, as dermatitis, where the mites and ticks fixate, an immune response that is mediated mainly by cells. An important sign is anemia and dysecdysis. as you can see in these pictures. And from those mites that are inside the or are mainly located in the lungs, we can see animals with respiratory symptoms or signs such as open mouth respiration, stomatitis, stargazing position, which means that the snake cannot breathe, so it will try to move its head in this position to be able to grasp some air, and most commonly pneumonia as a final effect. Other important side effects or deleterious effects are those produced when depending on the tropism certain species of mite have, they will locate or fixate in very sensitive areas. For example, vipers, most of them have loreal pits, which are some type of sensory regions which allow them to do a thermographic image of their environment. When the mites are located within the loreal pit or even in the eyelid in the eye, because they don't have eyelids, they can produce impactation. So most importantly, as I showed you before, we have a very big number or large number of species of acari that can parasitize herpetofauna. But in this sense, they are most of them are very specific to their hosts. So this will give us an important implication in the medical and veterinary concern that we can have given by the ectoparasites. For example, if there is an animal that has a parasite that has a high host specificity, as in this case of Pteridosomatidae, which are specific mites from geckos, we will have that the transmission of different pathogens or agents will be just within the species. Differently, for example, if you have a tick or a mite that has a low host specificity, for example, this is an Amblyomma sculptum, a very important, or the vector of Rickettsia rickettsii, which is the Brazilian spotted fever disease, which in this case is parasitizing tegu lizards, but of course it can also feed on humans. So this is important to be able to assess or to examine the vectorial competence or role to different pathogens of zoonotic concern, of course. So we have that the effect of the parasitism can be given also as these ectoparasites can act as vectors of viruses, bacteria, and protozoa. So here we have from the bacteria, mainly the Ixodida or ticks are more linked to the bacteria such as Coxiella, the spotted fever or Rickettsia, the Lyme disease or Borrelia burgdorferi sensulatu. Some also are linked to Thrombidiformis, mainly to Thrombiculidae, and Mesostigmata, mainly to snakes or to the group of reptiles, not so much to as a zoonotic concern, are linked to, for example, viruses such as the one that you can see in the video, which is a very important in the herpetological medicine, a very important virus, which is the inclusion body disease, which is caused by arenavirus transmitted by the Mesostigmata mite or Phoenis natricis, also known as snake mite. And this is a very critical disease because it doesn't have any type of cure, and the prognosis is very bad always, because once the snake presents clinical signs, most likely the animal will die given this disease. But Mesostigmata can also be mechanical vectors of some bacteria, for example, Aeromonas, and some protozoa. They can be they can have a passive transmission when the for example, for a pathozoon, when the animal eats the own or another reptile eats the ectoparasites. And Thrombidiformis, mainly Pterygosomatidae, this one from the geckos, are linked in this passive transmission to hemogregarines and other protozoa such as hepatozoon. So before coming to Italy, I was in Brazil, and my main focus there was to try to do an integrative herpetological parasitology applied to the acarina, or to the mites and ticks of reptiles and amphibians also I work with in Brazil. And in this case, the first thing that we need to discuss is how to capture the animals. I work mainly with synanthropic animals or animals that were brought to the lab. So the animal capturing, this was in Basilicata in a study we did with Borrelia from lizards. So one of the main ways to capture, for example, lizards is by hand, or for example, these are Lacerta bilineata or Ramarri, or lizard noosing. Lizard noosing is a way of fishing lizards because they do not respond to the nylon thread. So if you come closer with the knot, they will not respond. As you can see, for example, in this video, we performing a study in Filicudi Island in the Aeolian Islands, for which the animal do not, when they do not see this as a threat, so they can be captured without harm, and then we can continue to process them. After we capture the animal, or when we're going to capture the animal, we have to take into account certain situations or certain factors for when we do the handling and the restraint of the animal. So the first thing we need to do is the risk assessment. Of course, for example, if I'm working with a small lizard, the risks are scarce in the matter of some type of scar or some type of injury. On the other hand, transmission of diseases is possible, so we always need to consider the animal as infected or treated it with the proper security measures. But in this case, for example, a large squamata, a large lizard, an iguana, we have to assess the risk. If, for example, I'm working with this animal, I need to capture it to see if it had ticks. What should I see first? I should see that, for example, it can bite. They are herbivores, but they have very large, not very large, but very thick and very harmful teeth. So it's important to evidence where the head is to not suffer from some bite. And then most importantly, believe it or not, as these animals are, they live in the trees, they have very big claws, and they can be also very harmful when not treated or handled properly. But we do not need also to just think on our safety. We need to think on the safety of the animal, because obviously it's a stressful situation. So we need to see what are the things that could happen to the animal. So iguanas, what do they do when an iguana is generally a reptile, when it's stressed, or it seems that it's in danger? It will try to increase its size to show that it's larger. And iguanas have a very interesting behavior that they will try to whip with the tail. As you see the position that iguana is keeping, and this can hurt. But most importantly, not only it can hurt, remember that most of the lizards, they can produce a caudal autotomy, which is that they can free loose a part of the tail, and in this, the tail we keep moving, and then it can escape. This is important in lizards such as iguanas, because they can only regenerate the tail once or twice, and then obviously the tail would not be the same as it was before. Regarding snakes, we need to take into consideration these snakes that can be venomous. For example, this is a Micrurus surinamensis from northern Brazil, in Pará. And remember, I don't know if you know that there are a lot of species that produce Bayesian mimetism, which is that they tend to look as they were venomous. So there are a lot of false coral snakes. And not only it's important, but also snakes can be, depending on the species, aggressive. And in certain areas, we have the distribution. For example, here we have viperas in the different regions. which are also important to know how to handle and how to distinguish that they are venomous or not. Of course, size matters, as I was telling before, in the sense that for a lizard, a small lizard, we will need to take into consideration not harming the animal, be able to handle it properly. And with large animals, we need to understand what is the mechanism they have to defend themselves, so we can handle them and restrain them from harming us and harming themselves. So for snakes, we need to have a proper equipment such as hooks, thorns, thongs that will allow us to grab the snakes. Also, tubes are very important for mainly for venomous snakes, and bags to be able to handle them properly. So three main points to have into consideration when handling a venomous snake. This is Bothrops, I forgot the name, Mugini, lancehead snake. And we have to take into consideration three main points. Distance. A snake in this position, it in this position with the sigmoid form, can in fact largen its size 2 or twice the position it is. So we need to be from a certain distance to not receive or to not be bitten by the animal. It's important for this type of animals and for others, when other snakes, when you need to grab them or to handle them, to secure the head properly, which is with the hook in between the occipital region and the beginning of what would be the neck, in this case, where the vertebrae are. So there are very different ways of handling snakes depending on the initial assessment. For example, if a snake is non-venomous but aggressive, you can handle them with the hook, separating the head from distancing the head from your body. From non-venomous boids and pythons, you can secure the head holding them around, differently from vipers, because they can in certain way still hurt you with their teeth. And in some cases, when the animals are non-venomous and are easily handled, for example, this is Pilotes pulatus or Caninana from Brazil, we were doing a throughout examination for mites and ticks. And as you can see, the animal can be easily handled. As other animals that are not aggressive generally, as for example, Oxyrophus or false coral snakes, or some Philodrias also, which depending on the health, on their health and on their initial behavior, you can handle them sometimes properly or easily. Differently from venomous snakes, which you have to take into consideration that, for example, vipers have the ability to move their teeth. So the best way to handle them would be with this type of acrylic tubes that will allow you to see where the snake is, handle them, do the examination without risking any accidents, as you can see. So when we have the animal sedated or some animal that we can work on with no problem, it's important to examine the animal from anterior or cranial to caudal posterior, starting with the head, in between the skin, the scales, because mites and ticks are in between those regions. The gular area is a very important region, because as you can see, there's a lot of connective tissue in between the scales where the ticks and mites prefer, and also because snakes, actually, they have a ligament in their neck, which in some certain species, they do not move with the head in the ground. The head is a bit lengthened from the body, which means that this region is very protected for mites and ticks. Also, it's important, as I told you, for the entonicid or lung mites to assess the mouth part, the choanas and the glottis. And in between the scales, it's very important to see the connective tissue, because sometimes you can find the mites there. Also, just to show you the mouth, again, important. Why, for example, this is a study, a short communication study we did in 2018 from La Equator lineata in Basilicata, which we found that in the mouth, it had larvae Apixodes riccinus ticks. So that's why it's very important not only to assess externally, but the cloaca and the mouth, the oral cavity of reptiles. Afterwards, we can continue with the animal sampling, which for mites and ticks, the most important part is that scarification, which is the gentle extraction of the mites and the ticks with a needle or with a forceps. And in this case, we can take the mites without destroying them, with the whole structures, and then we can analyze them directly or molecularly without any problem. So you can see this is a wall lizard with mesostigmata mites, and this is a iguana with pterygosomatid mites. Also, see, this is a type of lizard from Brazil, which also, this is just to show you how this scarification can work. As you can see, it needs to be performed very gently, taking one mite at a time. And this way we can put them on the slide, or if we want, also in the alcohol to preserve them. From ticks, depending on the generally mites, the lizard will be affected by larvae and nymphs. And in South America, amblyoma, all of the different life stages will affect reptiles, so that we can take them with some tweezers gently to be able to collect also the hypostome of the ticks. Once we have, we can then process the samples, slide preparation to have better morphological details. We can take samples and perform blood smears from the reptiles to also see if they have some hemoparasites, and we can extract DNA from mites and the hosts, and also perform histology for just to see if there are some effects on the host. But with the DNA extraction, then we can perform molecular examination. Generally, when we work with philanthropic animals, the important thing is to, at the end, the animals go back to their environment with any problem. Then when we have the mites, we need to identify them properly using, for example, dichotomous keys. Original descriptions are very important. Types, for example, holotypes and paratypes deposited in different archaeological collections. And we need to use specific terminology, which I will not enter in depth because it's also a lot and very different from one family to the other. But just in the general concept, it's important to clarify the animal, the parasite. For example, this is a Ophioptes mite. Here, it should be the anal opening and also some very important seta and structures. So we need to clarify them with different methods, as lactophenol or lactic acid for a couple of days. And then we can see the mount the proper slides to be able to see all of the different structures that we need to be able to identify properly. We can also perform illustrations with a special microscope that will enable us to describe a new species, which is not very uncommon, and to be able to point out the specific parts that we use to diagnose that species. Also, something very cool and very important that we were able to do in Brazil was the scanning electron microscopy, which we were allowed to describe specific morphological assets that were only visualized with this type of technique. For example, this is a Jacobia harrisi, sorry. And you can see that here it has the respiratory stigma protruding between the natosoma and the coccyx. So this is something that is very difficult to see in the slide, but it's very well observed in the scanning electron microscopy image. For the molecular biology, what we can do is assess mites and ticks for their endogenous control and also for their barcoding, and then infer some phylogenetic analysis. And we can also, from the mite, ticks, and from the host, we can further on study important or vector-borne agents from both animals. So the techniques are various. We have PCR, conventional PCR, real-time PCR, which are useful in this case. And then with the purification and sequencing, we can then, for example, better understand if the parasites and the host had some type of important pathogen. In this case, when we studied Borrelia, we were able to observe it in the ticks, Ixodes riccinus, and in the lizards. So now, just to finish, I will show you some studies that we performed the last couple of years in Brazil and also here in Italy. This is one of all of the integrative acarology we try to apply to the epidemiological parasitology. This is one of the most important ones we perform, which is the we cataloged, identified, and see the preferred locations of different parasites, mites, and ticks from reptiles and amphibians in different ecoregions of Brazil. So as you can see, the green gets darker when we have more cases or more records. So the record, the place, the state that we found more samples was in Sao Paulo, of course, because of I was working mainly there. And we were able to then find different types of mites and ticks, even some that are not specifically parasitic as Oribatida, which are free-living mites. And we found it in different type of hosts, such as lizards, snakes, and even tortoises. We found some specific co-infections. So with the scanning electron microscopy, we were able to identify those of importance. And then the preferred locations in the sense of where the animals or the mites or ticks were localized. For example, in these Kenthoptics carcatra, they were in the near the armpits or in the hind limbs. And for ticks, that we find mostly in snakes and big lizards, they were located throughout the anterior portion of the body, and even here in the near the eye, of ticks. Afterwards, with this important study, what we could do was, okay, let's try to assess, or let's see what are the different vector-borne agents that these parasites can harbor or possibly have a role in their transmission. So in this case, as you can see, here are the states where we had samples, which are in light green, and those in dark green or darker green are where we had positive animals for different agents, mainly Rickettsia bacteria and Hepatozoan parasites, you can see here. So from the Hepatozoan, we found 3 types, and for Rickettsia, we had different types from the spotted fever group, or one Rickettsia from the ancestral group called Rickettsia belli, which has been associated generally to Amblyoma humerale and Amblyoma sculptum and Amblyoma rotundatum, which is a type of Rickettsia which is non-pathogenic, and it seems to be in specific ticks from reptiles. So we were able to construct a dendrogram for Hepatozoan in which you see 2 groups important. One was found in also mites from exotic reptiles as Pogona viticeps or bearded dragons, and from endemic reptiles as Phyllodes natereri. And then we have another group here, which was from ticks and from snakes of the Acre region. And this group was identified as Hepatozoan musa, which is one species of Hepatozoan already described from Brazil, from rattlesnakes. Finally, from this study, we were able to also reconstruct the phylogeny of infer the phylogeny of Rickettsia. And importantly, we found that not only ticks were associated to different Rickettsia, but also some type of mites, like, for example, Jacobella harrisi, which is a very specific mite from some type of lizards. So this opens the question on how the ecological pathways or relationships occur in endemic areas of different pathogens depending on the initial host or the proper host, and then the other feasible or sentinel hosts that they can be near or occurring in sympatry. So talking a little bit about the Italian context or the work that we developed in Italy, the first one was in 2018 in Basilicata mainly, where I participated of a type of Erasmus project called Global Doc. So we decided to work on a specific point that was Borrelia burgdorfis ensulatum, which was already described from the northern central region, from central region, yeah, from Tuscany, from Podarcis muralis, which is a very common wall lizard. So we wanted to see what was the distribution or the occurrence, in fact, here in the south of Borrelia. So then we published this study with reptiles that came from Puglia and Basilicata, and that were or synanthropic or were kept by owners, also from zoo animals. And we found a very diverse number of species, from pterygosomatid mites, these two up here, mesostigmata mites from Ophronisus, and then chigger mites or Thrombiculidae, and co-infestations, which is something very important because the co-infestations were with two possible zoonotic species of mites and ticks, which are Ixodes riccinus. It's a very important vector of different bacterial pathogens, and then Neutrombicula autumnalis, which can infest in fact mammals. And some studies have shown that it can possibly have a role as a vector of diseases. Other type of thrombiculids, such as Leptotrombidium, can be the vector of typhus fever or Orienta Susugamushi in Japan. So it is possible that Neutrombicula autumnalis could be also vector of some pathogens. So we wanted to see this, but in fact, Ixodes riccinus was the one that we found mostly positive for Borrelia. And the main Borrelia we found was Borrelia lusitane, which is a Borrelia from the group of Borrelia burgdorfis ensulatum that can produce clinical signs in humans, mainly from, was described mainly in Portugal, but also now in Italy. But what about Neutrombicula autumnalis was something that we were okay. It's possible that it can vectorize or it can have an important role with some pathogen. So recently, after coming to Italy again as a doctoral PhD candidate, and now as a associate, as a fellow researcher, we studied these two zoonotic ectoparasites to see if they have some role, and also reptiles, with rickettsia. So we recently published this article, very interesting article on the role of these reptiles and their ectoparasites with the epidemiology of rickettsiosis using a One Health approach. So what we found is that, in fact, Neutrombicula and Ixodes from different type of reptiles were from the spotted fever group, with the OMPA gene, we found Rickettsia monachensis, and with the GLTA gene, we found monachensis and helvetica. Also, other type of pathogens that we found, or agents that we found, were Candidatus allocryptoplasma, which is a type of anaplasmatachet that is not well known the pathogenicity, and some species of Ehrlichia. So as I mentioned before, we wanted to do, our objective was to show or to have a One Health approach. So we also assessed the seropositivity for different vector-borne agents of the people working or living near these reptiles. And in fact, it seems that in this epidemiological context, they can transmit or harbor different important bacteria such as Borrelia and Rickettsia, Coxiella, and Anaplasma. to finish, I will just talk very quickly regarding the work that we're trying to develop, or we are now, or my main goal as a researcher, which is with a scarce studied group, which are sandflies and their association to reptiles. So this is just to show you that vector-borne parasites, such as hemoparasites, protozoas, they have an immense diversity that I cannot even talk how many species. So this is just to show you one example of a very specific place, a point in the Amazon region of Brazil. They perform a study with lizards, and this is all of the type of hemoparasites they found, from Hepatozoon to Sauroplasma, Plasmodium, to Sarcocystosome, to Phallacia, to Garnia, Trypanosoma, Microfilarias, Trypanosomas, and Artifact. So it's such an immense diversity that I want you just focus on Trypanosoma, ones that can be zoonotic, and Leishmania. So for Trypanosoma, there are only scarce records of possibly involving monitor lizards that are a feasible food source for Glossina or tsetse flies, and they can be susceptible hosts for Trypanosoma brucei, which is the rhodesian sleeping sickness, mainly in Kenya. Finally, for Leishmania, well, just to do a quick introduction, within the Leishmania groups, we have two clades, the Leishmania clade, which is what the one containing the pathogenic, mostly pathogenic Leishmania, which is mainly uses reservoirs or host mammals such as rodents and canines, and is vectored by Phlebotomus in the Old World and Lutzomia in the New World. And then we have a sister clade, which is Sauroleishmania, which is mainly from lizards, and it's transmitted in the Old World by Sergentomia, the genus Sergentomia. But in fact, in the New World, in Central America and South America, some species of Lutzomia and other species can in fact feed on reptiles, but we don't know absolutely nothing regarding their importance or their possible role for the pathogenic Leishmania. On the other hand, knowing that Leishmania tarentule or Sauroleishmania, some species of Leishmania, occur in sympatry with Leishmania infantum and other pathogenic Leishmanias, we're now trying to assess this interaction. So for the sandflies, in specific epidemiological context, for example, lizards, as you can see here, live in sympatry or live near the Muretia secchi, where we put some of the methods we use to capture sandflies, such as sticky traps and CDC light traps, which are also located near in these specific places, are in shelters. And this is very important because what we're trying to assess is the sympathetic occurrence or interactions between Leishmania from lizards and dogs in endemic areas of canine leishmaniasis produced by Leishmania infantum here in southern Italy. So what we know is that Leishmania infantum, for example, which is the main species in Europe producing canine leishmaniosis and human and visceral leishmaniasis, it uses mammals as main hosts, and female Phlebotomus sandflies are the ones that vectorize this pathogen. On the other hand, for Leishmania tarentule, actually we don't know much about the cycle. So we don't know if actually the Serientoma minuta sandflies are the vectors, and we don't know the tropism it has inside the geckos or hosts. On the other hand, Leishmania, Sauroleishmania group has one species, that Leishmania adleri, which has been already identified as a possible causative agent of cutaneous leishmaniasis in Kenya. So this group, there is a wide area to study the possible implications of the occurrence of both species. And in fact, for example, in northwestern China, In 2019, they performed some molecular detection of Leishmania species in lizards and snakes, and they found that in that region where they didn't have the proper host, or there were not so many dogs, they found pathogenic Leishmania such as Leishmania turanica, Donovani, and Major. So we see here that we have two situations in which it's possible that lizards are reservoirs or reptiles are reservoirs for pathogenic Leishmania, and it's possible that mammals can also be exposed or infected by Sauroleishmanias. And what is the interaction? And this recent study also spurred our interest in this particular reason or a particular study, because recently also in Italy, Leishmania tarentulae was detected in humans, donors, in human blood, also, but not in dogs. So this study, what shows is that humans can possibly be exposed or infected by Leishmania tarentulae, but that is also seen in the sense that Sergentomia is one of the most abundant sandflies occurring, at least, for example, in Italy. And Sergentomia has been found already as a possible, at least positive for Leishmania infantum in canine leishmaniosis endemic areas. And also in another regions of it's also been found infected, for example, in Portugal. And in Sicily recently, it was found positive some individuals for Leishmania donovani complex, obviously Leishmania tarentulae, and Leishmania infantum. But most importantly, what we can see is Serentoma minuta is considered an herpetophilic sandfly, which means that it feeds mostly on reptiles. But this study, recent study, what it showed is that Serentoma minuta possibly can have a more anthropophilic behavior, feeding in, for example, in these different situations or sites. It feeds mostly on humans. So in this context, in the Mediterranean basin context, with these two species occurring in sympatry, there are a lot of questions that we want to assess and to try to answer, which is, for example, if lizards and other synanthropic reptiles can be natural reservoirs of Leishmania infantum. Leishmania tarentulae could infect mammals or other mammals such as dogs. And sandflies, are there real vectors? Or, for example, Phlebotomus can transmit Leishmania tarentulae, or Serentoma minuta can transmit Leishmania infantum. So there are a lot of different questions that we are starting to address. With one of our recent studies that we developed a duplex real-time PCR to be able to, in fact, distinguish these two types of species. And we established this protocol using these two types of species, Podarcis siculus and Tareotura mauretanica. So finally, the work or the studies that need to be digressed or further approached are those that remain within the reptile vector-borne diseases, which we have reservoirs, reptiles that mostly are synanthropic animals that can harbor a myriad of microorganisms, from virus, protozoa to bacteria, which some of them can be pathogenic to humans. And we have also the interaction with the vectors, many of them which have not been identified and which we don't know exactly the role. And most importantly, the zoonotic diseases and how this all interacts in a urban settlement or the human-reptile interaction. So this is most of all my area of research that I've been investing for the past year. And well, I hope you have liked the presentation. I didn't go more than the 2 hours. So thank you very much for your attention. I hope you liked it. If you have any doubts or you would like to discuss some of these different subjects or topics that we talk about today, you can always write to my email, or we can enter in contact and discuss about it. So thank you very much. Grazie mille.
Thank you, Jairo.
Thank you.
For a very nice presentation. And I think that we have a couple of minutes to some questions from the classroom. There is just a comment from our friend Marco Pombi from Facebook that has to leave, but would like to discuss later about this aspect. And I don't know if in the classroom there are any questions.
Yeah, I have a question. First of all, thank you for this wonderful seminar.
Grazie.
Very, very beautiful. And so, in your opinion, it makes sense to search for Leishmania tarentulae or infantum in turtles? And if yes, how would you proceed?
Yes, so the information we have on the possible reservoir role of reptiles is quite scarce. As I mentioned before, the only study that has found pathogenic Leishmania in reptiles is from China. They did a study first with lizards, and then they did another study with snakes. So I think there are no studies with turtles. But if we take into consideration not the animal per se, but the epidemiological context, I mean, for example, it's very common to have testudo hermani, testudo greca in urban settlements where we have dogs, where we have sandflies. So as I showed you in the picture, reptiles not with regard it's not important the type of skin. They can still be bitten, or sandflies can feed on them. So in my opinion, I think it would be very interesting to assess if turtles in fact could have a type of role as reservoirs. Absolutely, because for example, we know that the Leishmania tarentulae is mainly from geckos, but possibly we're trying to see if it can affect other hosts, other reptile hosts, because not only if it occurs in sympatry with other species, we want to see if it's possible that it can infect snakes or other type of lacerated lizards or other type of animals. With turtles, what I would advise you, for example, we have done some examination of some turtles, not much, I think 10, in which we took blood samples, prepare a blood smear to be able to observe if we found some amastigotes or other type of Leishmania forms. And then molecularly also we try to detect it, but thus far we didn't found in turtles. I would not discourage this does not discourage me, because as I told you, it's a very low number of animals, so it will be important also to see where they're from and to have a more representative number of animals to able to assess the prevalence if it's possible.
Thank you. There is also Simone that would like to make a question.
Yes, thank you for your presentation. Very, very interesting. And I have a question about the distribution of ectoparasites, the area of most incidence of ectoparasites. So do you notice an higher incidence in more anthropoid area, or in area or in a wild area?
Okay, I will divide my question in three, because you have to take into consideration their question. You have to take into consideration different factors. For example, the anthropic pressure. Yes, of course, with first the anthropic pressure, then the geographical area, and then the type of reptile and the type of ectoparasite. I can give you 2 examples. Here in Italy, when we performed a study to assess the ectoparasites of reptiles in Puglia and Basilicata, in Puglia, we didn't find ticks at all in the synanthropic lizards. And in Basilicata, yes, but that can be because of the distribution of Ixodes richinus, which is generally in high, in mountain parts. And it's believed that here in Puglia, it doesn't occur. Differently from Neutrombicula. Neutrombicula, we observed it, or we found it distributed thus far. Everywhere I have seen a lizard, 1 or 2 at least have ectoparasites. In the other study we did in Brazil, it was a bit different because what we tried to do was to understand or to highlight which is the preferred location within the host, depending on the type of parasite. So for example, ticks, where does a tick go in a snake? Mesostigmata mite, where does it go? So it depends on the type of tick, the type of host. For example, for turtles, for tortoises, which they have a specific tick. In South America, it's a species called Hyaloma humerale. And here in Northern Africa, we have Hyaloma aegyptum, and they generally will enter inside the carapace within the fore or hind limbs or in the neck. Differently from snakes that generally you have taken into consideration where the tick will prefer to be, in the sense that where it will be less exposed. So it's not that the tick goes and knows that it's in a good area, but it's because as the animal molts or shed the skin and moves, it will lose the ticks, and only the ticks that selected a proper area will remain in the body. The other thing is, for example, mesostigmata mites, they are not fixated on the host. So as I showed that Opistoglypha natris is the snake mite, when you found an infestation on an animal, they are very common in pet snakes. When you find an animal infested, if you do the count, for example, it has 10 females feeding right now, you have to take consideration that is like the 10% of the population, because they will go and each time feed, and then they will remain in the environment, and then they will try. They have positive phototropism, so they will try to go up and try to go to another region. That's why it's very common to find in collections of snakes that the infestation occurs very fast, because people don't realize that the mites actually are in the substrate or not in fact on the snake.
Thank you.
Another 2 questions.
We are very Simone. There is the email, the contact of Dr. Mendoza, because we are out of 10 minutes and there are other lessons. So I would like to,
Sorry.
to close the meeting, even it was very, very interesting. But maybe we can organize another with also your friend, because Jairo, Simone is a breeder of snakes. And so this is the reason why he's very interested in that. And we also have in our department some snakes, some boa constrictor and python. So many thanks, Jairo. And I hope to see you soon by person, and maybe here in Sardinia. And thank you for everything also from my students.
Thank you, Antonio, for the invitation, and I really hope it was interesting for you, the presentation. And of course, if anyone wants to discuss, we also have an interest on pet reptiles, because the information we have on this type of animals is also very scarce. So anyway, hope to see you soon also, if not here in Bari. Hopefully we can go there in Sardinia to know Sardinia.
Yes.
Not so distant future.
Thank you. Bye bye.
Thank you.
Bye.
Thank you.
Thank you. Bye.
Thank you. Bye.
Ciao, buona giornata.